Thank you for your introduction. Today I'd like to discuss on presentation entitled Integration of OMICS into Metabolic Flux Distribution by Complementary Elementary Mode Analysis for large metabolic networks. As we know, the integration of OMICS is now challenged to the elementary mode analysis because the metabolic network is large, then computation of all elementary mode is very difficult. So we try to calculate the first complementary elementary mode analysis method for the integration of the OMICS data into metabolic flux distribution. Content, at first I'd like to some background of our research and then specific some problems and objectives. Third in methods, we discuss our method and we compare our results and discussion with the others methods, and finally concluding remarks. So in the omics layer, and when we study in DNA in genomics, it's study on DNA sequence of an organism, and RNA is transcriptomics. Here is analysis in mRNA molecules, and proteins is proteomics, and last one is the metabolomics. So if we understand the network behavior, that means the phenotypic behavior of the metabolic networks, then we need to analyze the metabolism. And elementary mode analysis is very important, and essentially it's structural properties to analyze the metabolic behavior. So when we study some research concept here, in when we study the bioinformatics is known as the top-down, and here is the parameter values are estimated by reproduce of the biological behavior. And another one is system biology, is known as the top-down methods. And here the mechanistic model reconstruction based on the molecular architecture, and another one is bioengineering. Here is the research in the problem-based research, and here's needs to the some medical schools or the hospitals and the companies. And those are methods are analyzed by a computer, and our ultimate goal is computer-aided drug design or development, disease diagnostics or prevention and treatment, and the patient care. So in system biology, there are some systematic to analyze the large complex and data. Here is an example in the car and the human, and we want to compare the two systems. So there are some essential parts is run in the car, and also the some parts are the human body. So we want to understand the system. We want to repair when some parts are failing, and we want to improve the performance more. As for example, in human body, we need to study the how proteins and genes are working together, and thus this system is very complex in biological system. So metabolism is an important biological process to analyze the metabolic behavior of this complex process. And here is an figure. It shows that the metabolic different types of metabolic pathway map, but those are complex and highly interconnected to each other. And some problems in metabolic networks is some disorder is related to some disease, like as obesity, diabetes, high blood pressure, cancer, and so on. So it is important to understand properly of metabolic networks using elementary mode analysis at steady state level to study of the metabolic behavior. A simple example of metabolic pathway is first start the molecules A, and we get the products of D and C, and B and C is the intercellular metabolites. And reaction 1, reaction 2, and reaction 3 are catalyzed by enzyme 1, enzyme 2, and enzyme 3 respectively. So network-based metabolic pathway analysis are essential to analyze the metabolic behavior. It's focused on the two methods. Number one is elementary mode analysis, and number two is extreme pathway analysis. There are some difference between the elementary mode analysis and the extreme pathway analysis. I'm not going to details. In our research, we use elementary mode analysis, which is the minimum set of enzyme that can operate at steady state level. And on the other hand, the extreme pathway is the systematically independent subset of the elementary modes. And another important is that elementary mode coefficient, that means the quantitative contribution to associate this elementary mode, and it's made by maximizing a general objective function. We specify some problems. The ordinary and existing method is the number of EM is very complex to calculate the large scale data. If we increase the network size, then the complexity increases exponentially. And second one is the elementary mode can be described by many scalar product of each elementary mode. and the predicted flux distribution must be independent of them. And final one is many organisms still do not provide any specific biological objective function. So our propose is that we use several times faster and efficient elementary mode we call the complementary elementary mode analysis. First, we apply the EM decomposition, which generates the major elementary mode to complexity of the all calculate of the elementary mode. When we apply the elementary mode decomposition method, then some input flux are needed, but in our research, some input flux are partially unknown. That's why we use alpha spectrum, and it is computed when the flux is partially unknown. And third step, we use maximum entropy principle to optimize the quantitative contribution of elementary mode coefficient to the prediction of the flux. This is the general process of our prediction of the flux. We use metabolic path EMF and then the computer. We also use the genetically modified mutants in our study, and finally we get the prediction of the flux distribution. This is the flowchart of our TM analysis method. Here is the 3-step: first alpha spectrum, then EM decoms, and the last one is maximum entropy principle. And this implemented in MATLAB. There are different objective functions in the literature. First is linear programming, which maximize this objective function, but it's not a good choice to optimize the elementary mode coefficient, because the maximum biomass or the ATP production and can vary between different organisms. So we can use quadratic programming, which minimize this objective function, and it is suitable for objective function when the biological function are not defined. But it restricted for the small scale and the medium scale, but it also depends on the scalar product of elementary mode. So it's also not a good choice for the optimized elementary mode coefficient. Another one is enzyme control flux linear programming, which maximize and minimize this objective function, and its empirical version is not a theoretical background. And last one is maximum entropy principle, which maximize this objective function, and it is straightforward formula and theoretically sound background. And here is no need to the additional biological hypothesis or objective function, or it's not depend on the products of elementary mode. So in our study, we used maximum entropy principle to the optimize of elementary mode coefficient for the flux prediction. This is the simulation results. At first, we use a simple example model. Here we use the 7 reaction and 3 metabolites, as is the stoichiometric matrix. Row represent the metabolites and column represent the reaction. And here the steady state mass balance equation, we can use this equation to solve this problem. And the input flux V1 is 100 is fixed and other is the unknown. So we want to prediction of the unknown fluxes. So we simply use the alpha spectrum method to the optimize, maximize or minimize the fluxes, and we show only the results. We get the 5 elementary mode to analyze the result. Then we apply the maximum entropy principle to optimize the elementary mode coefficient. And also we calculate the quantitative contribution of the input flux using this formula. And finally we get the 4 elementary mode is enough to prediction the flux distribution. Here see the last one is elementary mode coefficient. That means the quantitative contribution of the last elementary mode is 0. So it's no need to the flux prediction for our next step. We applied our method in 2 large scale model. First one is 140 metabolites and 156 reaction, and second one was 140 metabolites and 157 reaction. This is the network map for E. coli. I use our research, and here we see that the first 2 model 1 and model 2, we calculate the 2 method. Ordinary elementary mode is CellNet analyzer CNA and M-tool, and we study our CA complementary elementary mode analysis. And the elementary mode matrix is approximately 100,000 elementary mode by the ordinary elementary mode analysis, but our method only 202, and the second model only 295. On the other hand, the ordinary elementary mode generates the 300,000 elementary mode analysis. So in our step, we calculate the necessary and sufficient complementary elementary mode analysis. The first model is the number of CM, and this is the prediction difference using this formula. And we see here the first model, only 29 elementary mode is enough to the prediction of the flux distribution compared to the ordinary elementary mode analysis. And this point, after that, the prediction difference is almost convergence. For the second model, we get 35 complementary elementary mode is enough to prediction the flux distribution, and after that the flux prediction is almost convergence. And here we show that the employed CM and the EM and their quantitative contribution to the input flux, and we calculate for the first model the 4 elementary mode is common for the both method, and here we calculate the contribution of the input flux. And for the second model, we get the 12 flux is the common in the both method. And we see here the quantitative contribution compared to the ordinary elementary mode analysis is larger than our method. We compare our results for the first and second model. Here, the ordinary elementary mode analysis use 100,000 elementary mode, and we only use 29, and that is enough for the flux prediction of the model. And the second model here, ordinary elementary mode analysis 300,000 elementary mode, and our method use only 35. That means 35 is enough for the flux prediction of these networks. We also calculate the genetic modification of flux, that means coupled with two methods. Number one is a modified control effective flux, and another is enzyme control flux. And we apply our method for the genetic modification of flux. We show our results in here. We see that for the two model, the ordinary elementary mode analysis and the complementary elementary mode analysis are almost same results. But here mentioned that the ordinary elementary mode analysis, this use elementary mode analysis, but our method we use only the 29 for the first model and 35 for the second model. For the statistical point of view, we calculate the correlation coefficient and coefficient of determination between the experimental flux and the predicted flux, and we calculate also the p-values. Here our hypothesis was that the experimental flux and the predicted flux are the uncorrelated. And we here calculate for the two model in two condition, wild type and the mutant type. And we see here the correlation coefficient and coefficient of determination is high, and p-values are significantly low. That means we reject our hypothesis. That means the experimental flux and the predicted flux are highly correlated each other. For the GMF prediction flux and the experimental flux, we calculate the similar correlation coefficient and the coefficient of determination and the p-values. And we see here the correlation coefficient and coefficient of determination are high, and the p-values are significantly low. That means the GMF predicted flux and the flux prediction of the experimental flux are almost are highly correlated. Here we calculate the calculation speed and the accuracy of our method. Here we use the two method for the calculation, and the ordinary elementary mode analysis by the CNA and the M-tool, and our method is CNA analysis. And we calculate the prediction error using this formula. Mention that here the ordinary elementary mode use the two steps. First step is elementary mode extraction, and the second step is flux prediction. And the CM analysis consists of three steps. First, we apply the alpha spectrum, then CM extraction, and the flux prediction by the last step. And here we compare the table. We calculation speed 100 times are faster than the other two methods. And finally, we conclude that we found 29 and 35, which is a small portion of the ordinary elementary mode analysis. That is enough to the flux prediction for the model 1 and the model 2. And the predicted both maximum entropy principle and genetic modification flux are by the CM was very consistent with by the ordinary elementary mode analysis. And finally, the CM analysis is useful, and which neither requires the initial generation of full set of elementary mode, or we no need to the any biological objective function to do the prediction of the flux, and which is often computationally demanding and memory improvement and reduce the cost of the large metabolic networks. This is the reference. I use my slide. I would like to thank the Japan Society for the Promotion of Science to support our research. And this is the Professor Kurata's laboratory member. We study here some PhD and a postdoc and some graduation student. And thanks for your kind attention.